So I wear a couple of hats, starting with this particular session as we get into the idea of research agendas and talk about some of the specifics perhaps in areas that we could spend some time working more, because this is going to be, as I was asked to do, looking at kind of where we put our resources and what questions we ask, an academic kind of research. For the first few slides, we'll give some examples. And then the rest of it, I'll begin to talk about the funding and what it is that it looks like from the NIH perspective. And I'll carry that over in the session this afternoon as well. I think some of these numbers might be interesting and insightful to you. So just a little bit of background again to summarize a little bit from yesterday. When we start talking about what kind of research is needed to do with this particular viral agent, we're talking about a very specific kind of virus that is really difficult to work with. Compared to other infectious agents in the human brain, for example, JC virus turns out to be quite a tough critter to work with in the laboratory. And I think that we have to understand that. And consequently, I think that the work necessarily in progress is a little bit slower than some of the other viral agents that find their way into the brain. The biology of it, the molecular biology of it, the clinical aspects of it are really a little bit more difficult. In order to be able to accomplish that, I think then we need to, as the previous speaker had said, begin to collaborate, coordinate some of the technologies that we develop for this, maybe develop the concept of team investigations that direct translational work. And I think certainly our current director of the NIH, Dr. Francis Collins, puts a high value on translational work and translational medicine, and we appreciate that back in Bethesda. So some research areas, we touched on many of these yesterday, but we'll talk about them and flush them out a little bit more during the course of today. We haven't spent a good deal of time with this virus over the years looking at molecular genomics. We heard a good deal yesterday about the number of variants we find, the genomic variants of this virus. Do they have significance? How many more are there? Which ones should we focus on? The proteomics in terms of just the kinetics of synthesis of these particular virus proteins. We haven't spent that much time on it either. Viral gene regulation is dear to our hearts in the laboratory back in Bethesda because this virus has such a narrow and specific cell tropism, very different from other DNA-containing viruses that cause neurovirulent infections. Why, for example, that it specifically targets macroglial cells in the human brain, and with the exception of the observations that Igor Korelnik has talked about in this novel, insightful infection in cerebellogranular cells, for the most part, we never see JC infection in neurons in the cerebellum. Again, going back to one of the points that I had made yesterday morning when we started, I think we need to know more about the direct links that there are in immunomodulatory therapies and why it is that PML is this adverse event and essentially none other. And nature is trying to guide us there, and I think we have to really understand a little bit more about the physiologic effects of some of these immunological therapies and just the host response that we all have to this virus, globally distributed within the population. The majority of individuals, take your pick what percent you like to say has been prior exposed to this virus. What does that mean? What is really the host response to infection? And of course, we talked again yesterday a little bit about drug discovery, and is there a relevant cell culture model for this virus, given the fact that it has such a narrow tropism? And I'd just like to show you what it is we use in our laboratory in Bethesda, and how we like to deal with this to get to these particular issues. At the top of the screen there is, here is, we've identified it, isolated, purified, and culture from the human developing brain at various gestational ages, human neural progenitor stem cells. We have the ability then, having done that, to be able to direct their lineage differentiation in different cell types. So if we choose to make astrocytes, then we simply give it the kind of cues that's necessary to be able to do that. in a temporal period of time of about 3 weeks or so, we get 100% population of GFAP positive, professional looking and acting, biochemically acting astrocytes. JC virus grows like gangbusters in these cells. If we initiate infection, for example, in this population of progenitor cells, and then we move the directed lineage differentiation into the astrocyte population, the virus loves that cell. It just grows like crazy. That gives us the opportunity to be able to go ahead and then query that pathway to say, what happens? What are the molecular factors that allow that virus infection to take place? We can do that through genomics. We can do that through proteomics. We have a variety of different kinds things we can do. If we instead move that direction into a neuron by adding these particular kind of factors, BDNF, PDGF, neural stem cell factor, nothing happens. Absolutely nothing happens. And we understand one of the reasons of that is because these cells, neurons, particularly as they differentiate from a stem cell to a neuronal cell, don't have those kind of molecular factors which recognize the viral genome, and consequently nothing happens. Even more interesting, we can transdifferentiate the astrocyte into the neuron, and we can obliterate infection. We had thought we were getting really clever, and we can make oligodendrocytes, the target cell in the adult brain, of course. And we thought, well, that would be just wonderful because there we'd have the real target cell. And so we initiate infection, we move that infection into the oligodendrocyte pathway, we get very little infection, which is a surprise to say the least. We understand the reason of that is the fact that these are immature oligodendrocytes. And again, they don't have the complement of molecular factors which is required by the virus in order to be able to grow. We could do a similar kind of experiment if I just had a slide that had hematopoietic stem cells here and move them into various lineage differentiations, either to monocytes or lymphocytes. So this is the kind of biology that we like to use that's a relevant cell culture model for this. And it's not that difficult once you've worked out all of the pathways to be able to do that. Just as an example of perhaps one way to look at this as a research agenda and how to approach this. We also happen to think perhaps that the time is right now to take a look at the potential for developing perhaps vaccines. Peptide vaccines might be useful. We all are beginning to use VLPs, virus-like particles we talked about yesterday. We happen to have had the opportunity to work with our colleagues in the Cancer Institute, who are the ones that developed the papillomavirus VLPs that gave rise to the current vaccines that are globally used. So we learned a good deal from the technologies of working with Doug Lowy and John Schiller on that particular part of it and just transferred that to JC. And we could talk about that later as we get into maybe some of the afternoon discussions. But in order to be able to do something like that, you need to have an animal model. And of course, that is one of the big blockades that has been in this field for as long as I have. And I won't tell you how long that is. So we need to be able to do that. And, Igor had talked about the attempts that were made before using non-human primate models. And this is two of the reasons why I left Chicago to go to the NIH a long time ago. When the infectious diseases branch in my institute asked the question, what is this JC virus? And they were very good in developing non-human primate models for many other kinds of neuroinfectious diseases. They began directly intracranially inoculating virus into different species of non-human primates. Old world monkeys, I can tell you that there is no macaque that is around that likes JC virus. There is nothing we tried to do with JC, and Igor again mentioned one of the constructs we had made using an SV40 promoter vector that the rhesus macaque, when we put it into those animals, it did absolutely nothing. I mean, the virus did absolutely nothing. Unfortunately, in trying to develop a lytic infection in oligodendrocytes in a non-human primate, in New World monkeys, like these owl monkeys, named obviously because of their phenotype, instead of getting a lytic infection, what happened is you get tumors, and you get a malignant astrocytoma what's called a GBM, a glioblastoma multiforme. It is the absolute perfect model for a GBM. Histopathologically, temporally, in terms of the way that the tumor develops, it is an excellent model for GBM, but not for PML. Part of the problem with using this particular model, however, is that it takes anywhere between 18 and 24 months to get the tumor, and the animal succumbs to the oncogenesis of this particular tumor in a very short period of time. So it is very long-term, and it is very expensive, and it's difficult to manipulate the parameters of infection there. I still try to tell my colleagues at the Cancer Institute back home that this still remains to be the best model for GBMs that you could ever have. So we don't have a good non-human primate model. We don't have a good animal model at all for this, and I know that's something that the field would benefit from enormously, but it has not been because we haven't tried various kinds of ways of doing this. So let's take a look now, and let's put on the funding hat to some extent, and the research areas that are recognized by the NIH in terms of where these dollars seem to go, what actually is happening on the ground today. And on the column here on the left-hand side, you're just looking at the ways that research here has defined in terms of where these dollars go. Epidemiology and pathogenesis, molecular mechanisms and disease pathogenesis, again, therapeutic development and vaccines, the cellular response to infection, the structure of the virus, the clinical presentation, diagnostic features, the immunology and the virology, and this is taken from public reports. So I'm not speaking out of school or looking into the locked cabinets back at NIH here for information. And over the last 3 years where these data are available, you can see that the numbers of funded projects in these particular areas. So if you were to ask the NIH what is the research agenda there, these are the areas that come out. This is the way that we accumulate this information to say, you know, where are all of our dollars going? 24 projects. Let's contrast that in terms of what those dollars mean in terms of funding. Looking at those budgets, 2008, 2009, 2010, the low number is the dollars that the NIH spends on JC in all aspects of that. A couple of things to note. It's pretty much the same for all 3 years. About $10 million, a little over $10 million per year, spread out amongst all the institutes at the NIH, which is comprised of 27 institutes and centers. 24 of those are funding agencies, funding centers, and at an institute like mine, for example. If you contrast that with another common distributed virus in the human population, that's a DNA-containing virus, herpes simplex, it's quite a bit of difference. Now, people could argue that, well, herpes simplex, the incidence of morbidity and mortality with HSV is vastly more than JC, and you could make that case. There is a drug, however, as all of you know, that could be used in herpes simplex infection and other family members of herpes viruses. But interesting to note here is that the funding, however, for HSV, it continues to go up, where the funding for JC and PML pretty much has remained the same. And I think we have to ask ourselves why that might be the case. And let me just finish this, and we'll go on to talk about this again this afternoon. Funding is one of the issues. I think to more quickly advance research here, I think we need more formal support. Certainly it has not been for lack of trying back on campus. We need to have collaborations. And I think we need a infusion of some new ideas. I think this is an absolute critical point. We need to recruit new principal investigators from different fields into this field. And I think what we need now, as we begin to see PML that is in other underlying disease areas, then let's get some of those folks to work on this. We need people who are in areas of rheumatology, more immunologists. We'd love to see some basic neuroscientists who are glial cell biologists. I'll be going to Prague in September. I've been asked to put on a workshop there in the Euroglial meeting. And we're beginning to make introductions into those particular investigators in those fields, the Society for Neuroscience, to say glial biology can learn a good deal from those kinds of agents that specifically infect those cells. Because then you could query that and say, well, how does that cell respond to an infection? You can learn something about that using that kind of approach. And that's, I think, the way we need to think. We need to know much more about the genetics, both of the virus and of the host. And we need to get individuals who are committed to neuroinfectious disease programs, and that will all help to advance science. That will help to inform medical research for this. We'll continue to talk about this again this afternoon. Thank you.